out there in existence and used quite commonly. So next slide. So for the particular case of stirred tank reactors and cultivated meat, so there's a number of different starter cell types under consideration for cultivated meat, but a large grouping of them are what's called adherent, which means they must adhere to a surface to grow, to proliferate, and all that kind of good stuff. And so one way this is sort of achieved in these stirred tank bioreactors are using these small little bead-like objects called microcarriers. And there's a schematic of this process on this slide, and you can see all the cells that are adhering to a small little sphere. But the problem with this is that it renders the attached cells more sensitive to potentially damaging shear forces and turbulent flow. And then at larger scales, you're going to have to mix the system more vigorously to achieve the kind of homogeneity that you need. But that, in theory, can create more damaging shear forces. And so having these two kind of limitations in mind and knowing there's a trade-off between stir speed and shear forces, it kind of begs the question, well, how do you scale such a process? And one way to do that is to build a model, which is what the last part of this slide is trying to emphasize. So next slide, please. And so this slide here with all the little spaghetti wirings and whatnot it's not actually a microcarrier system, it's a microbial system, but it illustrates the point that, bioreactors, which have a lot of control, are quite complicated. So in this case, there's a whole host of different toggles or elements of the system which you can toggle to optimize the output. But if you wanted to create a model to encapsulate all these different aspects, that's going to be complex, and switching one element of the system you're going to have to understand how that impacts another. And so to create this sort of base model, it seems more important to start with something much simpler. So next slide, please. And that's precisely what we did for this proof of concept work. And so I had identified this older paper from 1987 by an MIT researcher, and the very simple bioreactor design that we considered is here on the left. And it's just simply a spinner flask with a little stir bar in it. And there was obviously cell culture media in there, and then the cells bound to microcarriers, and it was kept in an incubator. But what this paper did, kind of in isolation, was look at stir speed and then cell growth. And this seemed to be a great target to unite these two modeling platforms that we were interested in. Computational fluid dynamics, which could very accurately model stir speed and all the fluid flow components of the system, and then agent-based modeling, which could understand the cell growth and all the sort of forces that the individual cells experience. And so we took all the information from this paper from the 1980s, and we used that to build this foundational model, which was much simpler. And that's the end of my bit.
So hi, I'm JiaJia. I will introduce you, as kind of Greg mentioned, our first step of the modeling process is to develop a computational fluid dynamics model to simulate the flow field in these simple bioreactors. Next slide, please. So let's first look at what's the computational fluid dynamics. So computational fluid dynamics is also known as a CFD, which is a very kind of well-developed branch of the integrated disciplines of fluid dynamics, mathematics, and computer science. So CFD is kind of a general approach that uses numerical analysis and data analysis to analyze and solve the fluid flow in different types of bioreactors. And the CFD has already very well known to use in the different applications, such as to design the airplanes, the automobiles, in terms of to understand how the different equipment or different transport system will behave in the air flows. And similarly, actually, the CFD is also used widely in the bioreactors simulations to understand how the flow behavior in the bioreactors. Next slide, please. Then here, actually, just as Greg mentioned, our CFD model tried to simulate a very simple case from the published paper, Graham 1987 paper. The first step of our CFD model, which is to use their reported geometry, and we create the geometry in our software, and the software we are using here is a finite elements-based commercial software console. And also there are other types of software available, commercial software available, such as Fluent, which is using the finite difference approach. But here we are using console, and different methods, they will solve the CFD problem similarly. So after we develop the geometry, then we discretize the whole geometry to be millions of elements. This elements is kind of like a small parts of the geometry. And on each element, we may have multiple nodes. So we can calculate or we can simulate the flow behavior, like the velocity, the shear stress, etc., on each one of these millions of points on the nodes. And we are using this package to solving the different type of governing equations on each one of these nodes. And these governing equations generally for the fluid flow, which is Navier-Stokes equations, as shown on the right side of these governing equations. Next slide, please. So for all these models, for the CFD models, we actually can using the different solvers to simulate the different scenarios. For example, on the left side, we can simulate the steady state flow, which means if we mix the bioreactors for enough long time, how the flow field will behave inside of the bioreactor. So the velocity may change periodically at the different locations, but generally this change will not change with time. So that's called the steady state flow. We also can simulate the transient flow, like at a different location and a different time in the bioreactor, we can predict how the flow behavior behave inside of the bioreactor, like what's the velocity, what's the shear stress, etc., all this kind of stuff. So next slide, please. So in our model, next slides, please.
Okay, I did advance the slide. You should be seeing effective impeller speed velocity.
Okay.
Is that what you
see?
see?
I see it now.
Okay.
So in our model here, for this simple model, what we did is actually using the steady state model, because the advantage is that it can predict the results quickly, because it's a proof of concept model, and we use a steady state condition to simulate the fluid field. And we evaluate the different velocity, the different impeller speeds on the flow behavior. So here shows two different impeller speeds, 60 RPM and 240 RPM. We can see that the velocity profile looks very similar to each other, but if you see closely, actually, if you see the legends, the color legend of these two files, they have the quite different the velocity, the magnitude of the velocity. Okay, that's outputs from the CFD model. In addition to this velocity, we also can simulate like the shear stress, the dissipation energy, etc. All the different type of parameters can be predicted by this CFD model. And for the next step, Jaro will explain how the ABM model will use the CFD model results in the biological process modeling.
Hi all, here's Jaro from the Netherlands. I'm affiliated with the University of Amsterdam and the Netherlands eScience Center. And first of all, so I'm going to talk about agent-based modeling part of this proof of concept study. First of all, I want to mention that most of the credit goes to Boris Egler, who did most of the work on this, but he sadly couldn't make it to the meeting. So next slide, please. So a bit about agent-based modeling, in short, ABM. So agent-based models, in agent-based models, one simulates the actions and interactions of autonomous agents to assess their effects on the system as a whole. And specifically, emergent effects are very interesting. So, for example, cell sorting in the image you see below, which simulates tumor growth, for example, with the Bayesian framework. But it's also used in other biological systems. For example, a study from the University of Amsterdam, where they modeled the gastrulation of a sea anemone during its early development. So the point is that you simulate agents which are autonomous systems that can interact with their environment and other agents. But they are autonomous themselves. And it can, for example, also be applied in many different fields, like you see the image of the evacuation planning, where they modeled a crowd of people wanting to leave the room through one exit. And the output is how many people actually left the room and how many were injured. And it seemed very unlikely, but apparently, if you put a column, which you see in the bottom image, just in front of the door, more people will survive and less will get hurt in the end. So this, in the end, was used to optimize emergency exits by putting a column just in front of it. So next slide, please. So we use agent-based modeling to model the muscle cells and the microcarriers. So we have two types of agents, cells and microcarriers. And cells like to adhere to microcarriers and to each other. So all of these agents have several characteristics and can be in a certain state. And they experience forces by each other or their environment. For example, deformation force by each other. If they get too close, they get squished together and they repel each other. Or by the fluid flow, they experience a drag force. So we took the fluid flow field from JiaJia's CFD simulation, and we put all of these little microcarriers with cells on them in this fluid flow, and let this run for some time. And you can see the result of that in the video. So next slide, please. So here we have the results from the simulations we run. So on the left, we have these two different impeller speeds that JiaJia showed. And you see that with a quite slow impeller speed, we have faster cell growth, of course, because there's less stress on the cells. And with a faster speed, we have more stress, so also slower growth, because these cells get stressed and proliferate slower. But maybe a more interesting result was when we started to tune the stress parameters. So these cells, they experience force by either the velocity field or each other. And this results in a stress in the cell, and we made it such that these cells, that their proliferation rate depend on the stress they experience. So we have a stress parameter, and when they experience these forces, the stress parameters will define the proliferation rate of these cells. And you see, if we tune up the proliferation rate, the only thing that matters is actually the fluid flow. And you see almost a straight line. If we make it a bit lower, this parameter, you see at some point that there's the slope changes at some point. This is an interesting result, which happens when the microcarriers fill up with proliferated cells, and these cells start to experience each other's presence, and they start to push into each other, adding an additional stress to the fluid flow. Next slide, please. So this approach shows that that our approach, well, sort of works. Qualitatively, we see the same results as we had in the Krugman paper that Greg showed. But there's, of course, much to improve, because this is a really simple model, and we can add lots of different additional states or interesting effects in these agents, these cells. For example, we still need to add gravity to this equation, because that will pull the microcarriers down. Collisions between microcarriers could result in cell transfer from one to one bead. And there's, of course, much biology that we didn't model here, but which could all be implemented in the cell, so that the shear stress will influence the gene regulation network or accretion or nutrient consumption in these cells. But thing to mention is that if you add more complex models or add more stuff to the model, of course, this comes in with a cost on the computational side. So we need to decide which ones to add and which ones not to add. And that's the future perspective for this this part. So then I'll give the stick to Simon again.
Sure. So so you've seen the description of the setup, the scenario, how computational fluid dynamics results in these velocity fields, and agent-based modeling slurps up that information to predict the outcomes for the individual cells. So I wanted to talk about what the software pipeline looks like and where you can get the source code, show you kind of how the whole thing goes together to produce a like a demo, an interactive demo you can try, and quickly go over some possible application areas. And then I'll talk very briefly about the GFI grant. So next slide, please. So to begin with, we have this bioreactor geometry, which is generated by some domain expert. Well, creates a beaker or whatever that bioreactor might be, a tube, and feeds that into CommSol. Next slide. Then CommSol generates that fluid velocity field that actually has to be turned into a structure that the agents can find themselves in. They need to locate themselves in some sort of structure to figure out what the velocity is at their particular point, because as JiaJia mentioned, there's a mesh that CommSol uses. So there's software that's available for that. That feeds into Biocellian, which is an agent-based simulation modeling and simulation system, attuned to the needs of biology. That also has to take into account cell-to-cell biomechanics. That information has to come out of papers and resources, domain experts. So that's another input to the pipeline. And that microcarrier, ABM microcarrier model can be found on GitHub as well. Next slide. Okay, so the idea is that we're building out this pipeline. It's the software stuff. It doesn't know anything about biology per se, or about bioreactors. That other stuff needs to be fed into it, and that's where there can be proprietary information. So that's the pre-competitive technology is building out along the bottom, and then the technology that's specific to the problems, specific to companies, and so on could come in. Next slide. So finally, the Biocellian simulation spits out the cell microcarrier counts, their positions, their stress numbers. And again, that has to be pre-processed before it can be used for visualization. So there's some other software that's available on GitHub for that. And then there's an interactive visualization tool that we built, also available for download on GitHub. And we'll do a quick demo of that. Next slide. Okay, so you can go on our website, you'll find this. You can run it in your browser. There's a browser version. You can zoom in on this, on Proen's stir tank reactor, and you can see the microcarriers floating around with cells attached. There's a number of different things you can do, like you can type F to focus on a particular microcarrier, like that. You can jump from microcarrier to microcarrier, zoom in on it, and see the stresses as they change. Red means higher stress, blue means lower stress. as it floats through. So that can just give you some idea of what's happening as another means of feedback besides just graphs. Not need to run it again. Next slide. So possible application areas. You could think about maybe in the future looking at how to look at different tank sizes and shapes and rotors and different kinds of bioreactors, looking at the stresses of the cells, how mixing occurs. We're not doing any mixing here in this example. We don't even have any media in this simple scenario. People have talked about looking at different types of microcarriers. Some of them, maybe changing the microcarrier shape would reduce the stress on cells. Some people are looking at even microcarriers that are soft or edible. The bioprocesses is another thing people have raised as a possible application area. There's a whole process you go through. Some people try changing rotor speed, some add additional microcarriers, and so on. So looking for an optimal schedule. That can also be modeled and simulated. And of course, the scale-up issue of how large can you make a given bioreactor design, and those bioprocesses, how do they need to change as you scale up without having to run so many laboratory experiments. Next slide. So so that was the proof of concept. That was this past 6 months of work for our little team, sponsored by Merck. And going forward, we have, in addition to Merck's continued sponsorship, a grant from the Good Food Institute to look at novel designs, not necessarily stirred tanks, but we propose to look at rocking bioreactors, hollow fiber bioreactors, continuous surface bioreactors, and see how our pipeline needs to change for those. See how well we can characterize what determines their performance and efficiency. Do our best to design some multi-scale models similar to this one, and see what we can do to analyze the designs through our models. So next slide. I think that's it then. Thanks.
So now I'm going to stop share, and let's just take a few minutes here for any questions that might have come up for you all as you were listening to that. And my suggestion would be to put your questions in the chat. And even if it's not like a burning question that you want to have immediately answered, putting your questions in the chat, and I can see there's also quite a bit of discussion actually that's been happening as well. That's really helpful because we can capture all that as an artifact to understand what our community, what this group of people wants to look at, what questions are coming up, those sorts of things. So go ahead and think about what questions are pertinent, what's interesting to you at the moment, what's popping, and if you can, find the chat and type in your chat, and our team will, as they see questions roll in, take the next few minutes to answer some of the burning ones. I see a question already about the presentation. Yeah, we're recording the presentation. We'll distribute it, including the slides and the video. And yeah, links to GitHub. They're currently in the presentation, but obviously you can't cut and paste from there. So we'll be sending links along also. So I'm seeing a couple questions coming in that I can't answer because I'm not the right person to answer these questions. So if anyone in the team wants to pop in.
So I know for the agent base, so answering this question about the computational cost limitations when adding metabolism to the agent, the time to compute metabolics inside of the agent, of course, depends on how that's represented. But if you imagine it as basically consuming nutrients from the outside and secreting waste products outwards, at least those costs tend to rise with the number of molecular species because we have to solve differential equations across the whole space to determine how those molecular species propagate. So if you have a media with 700 components, it's going to be really slow. If you've got a media with just a few components, or there's a few key components and oxygen, then it's quite doable. We've done similar kinds of things in other models.
Unless one makes very restrictive assumptions like linearity and steady state, in which case you can use like flux balance analysis and use and have very large models.
So to Jed Johnson's question, does the model incorporate changes in mass and size as the cells proliferate? The cells do grow. Right now, gravity hasn't been added to the model. Obviously, that's a that's like a short term and fairly straightforward change. But the cells do grow as the simulation continues, and when they proliferate, they shrink back down.
Yes, actually, currently proliferation is modeled as when a cell grows a certain size, then it proliferates.
Right.
Yeah, like yeast cells.
Mm.
And I see a question from Maria. Can this application be modified for bioreactors with another design? And yes, it can. I think that's a significant part of the goal and of the 2 years of work that we'll be doing as part of the GFI grant is testing different geometries. And maybe you're aware of geometries you'd like to see tested. That's definitely an area that different geometries for different groups and different applications can be sprouted up here in this community, and we can look at that in different project teams.
There's another question about validating models from Leo. Maybe I could take a stab at it. I think the question is whether one will need to validate models using physical experiments. And I think the answer is yes. It will require physical experiments and specific experiments that are designed hand in hand with the model. There are certainly other data sets available on growth curves and cell growth for various cells and under different conditions, but those may not be easily generalizable. So I think it will be important to have experiments along with the modeling.
Yeah, maybe we have time for one more. I just want to answer, there's a couple questions that are coming in that are around things like what features are we interested in prioritizing, for example, or what kind of parameters are needed? And actually, those are questions that are really useful to capture and discuss in our next breakout session. And our plan is to, currently we have a team that's working on this POC, and we have some plans of how we want to approach the next phase of work, especially when it comes to GFI. But this community is a great example of, we need the wisdom of everybody in the room to help guide the effort. And we kind of see it as a tree. So right now we've kind of planted the trunk, and now there's a possibility that different branches of research, different branches of application can come in. And the invitation is for those of us in the room with domain expertise or modeling skill, capacity, et cetera, companies that have different problems that they could maybe see this POC relating to in their own particular bioprocess, et cetera, to come together. And that's a big part of what the CMMC is. It's like a scaffold where all of these different parts can adhere and do work together. So is there any other question from the team that needs to be answered that feels burning before we move on to looking at that? I see one with Clarice. Are you planning to collaborate with clean meat companies to get data on muscle stem cells? The POC, as it's currently has been informed by conversations that we've had on an ongoing basis with folks in industry. And we have a number of those who are contributing to the ongoing development.
This last question, what densities of microcarriers can you test to? Currently, it's very sparse. And one of the big pushes that we're making is to be able to scale that up from seeding all the way to maturity. And yeah, right now the models are too simple to be said to be any particular species. That would be the kind of the green stuff that gets input into the agent-based model. We'll try to support whatever features are needed to differentiate support the different species of cells, whether they're meat or chicken. Which, I mean, these were FS4 cells that were used in the Crowan paper, human cells.
So, and just one quick, maybe low-hanging fruit question is, are these models being developed as species agnostic?
Right, that's what I was just answering.
Oh, sorry.
Yeah.
That's the non-scientist.
The answer is yes. Yes.
I heard green stuff and then I got caught up in
no.
that. Awesome. Okay, so let's move. So what I'm going to do now, I'm going to share my screen and we're going to jump into some of these discussions. And we have a group process that we're going to run that is going to involve 3 rounds of conversation. Each round is going to be 15 minutes long. We're going to have groups of 5. And of that group of 5, there will be one person who's assigned as the table captain. Those people already know who they are. They're members of our steering group and POC group. And that table captain's job, and I'm giving them the nudge and the wink right now to maybe go away from the presentation and open up a fresh Google Doc or whatever technology you prefer. Their job is going to be to capture the conversation, to basically provide a record for it that we'll be sharing out amongst everybody. The table captain's job is to keep those notes, share their screen, and then when we do the next round, that table captain will essentially stay at their virtual table, and then new folks will be randomized together. So you'll be in 3 different groups. You may have some of the same people, you may have totally different people, but they're all going to be discussing 3 questions in sequence, and then at the end of that, the table captains will give us a brief report out. And the purpose of this is to, one, for everybody to meet each other and just enjoy meeting people maybe that they never have, to get people with very different kinds of expertise in the room thinking about the same problems, to answer any questions, extend our thinking, capture a lot of data, and generally just generate a lot of fresh material for the POC. And just really quickly want to reiterate that the reason we do that is because we all who've come together to form the POC, we have a very narrow point of view, our own. We've been working together now for several months. It's really time for this thing to expand and to get fresh energy and input into it. So, that's what the intention is of this particular exercise. So, yeah, so one question is, like, maybe you don't know anything about bioreactors and you're wondering, well, how can I? This is a great opportunity to ask questions. It's a great opportunity to learn. So if you don't feel like you can contribute to the discussion from an expertise standpoint, this is a great opportunity to just absorb a bunch of new information and learn a little bit more about the field and what's going on there. All right, so the first thing I'm gonna do is start randomizing us into our groups. And I'm gonna share that first question. You can start thinking about it. The question is, what can you imagine doing, what can you imagine doing with this POC-based technology in the future? What possibilities does it open up? And that can also include things like, well, what sort of things would we need to figure out in order for it to advance? So this is, it's a little bit difficult to share screens when you're in a group process like this. So you might scribble it down on a notepad, just take a second to scribble it down and then start to think, maybe write some notes yourself, how you would approach answering the question.
Sorry, Chris, what are we doing right now?
So right now, I'm randomizing people into breakout rooms, and you're writing down the question because you won't have it visible when you go to the breakout room.
Thank you.
Okay, so I'm going to open the rooms. You're going to have 15 minutes, and I'll let you know when we're back. And you should be seeing an invitation to join on your screen. I'm seeing a few folks who haven't moved yet. Great. So I'm seeing Steve and Rob and Devin. You guys can probably have an invitation somewhere on your screen asking you to join a breakout room.
Hey, Chris.
Hey.
Our group doesn't have a captain.
Oh, okay. I think Dario is supposed to be your captain. Oh,
He's
no.
not
Sorry.
there.
Hold on. Let me see. Well, I think we had intended JiaJia to do that, but if he's not comfortable doing that, can you do it?
No, I cannot. And I don't know that he is not comfortable. He's just not aware.
Okay, I'll jump in. I'll jump in and do it.
And Chris, the reason I cannot do it is because I cannot type, simply.
Oh, okay. I'll come in and then jump to it.
Okay. Can you put me back in that room? Or no, never mind. I can do it myself.
I think you can. Yeah, there you go.
Hey, Greg, it seems like it's two of us together.
Oh, that's it.
Yeah.
Okay.
Come on, take the lead, or shall I?
Oh no, I haven't randomized. We're all coming back to the same room.
Are we all in the main group?
Yeah.
So we'll
okay.
always come back to the main group.
Yeah, we'll always come back to the main group and then
Okay, got it.
and then back. So our second question, you can read it here. What next steps are needed now to move the current POC model forward? What's the most relevant or urgent? Simon, can I call on you to just reflect briefly on what stood out to you from that first round?
Sure. Yeah, sure. So we had Jed in our group who works for Matrix Meats, building scaffolds for other companies that want to grow meat. And what he said would be valuable is being able to predict the number of cells per what's essentially a scaffold microcarrier. So that would enable them to tell people how many of these they needed to buy. They could say, well, here, use this one, it's this size, it'll grow, you need to seed it with this many cells, and this is how much you'll get out. So in order to move that forward, we run into this challenge that we're already facing, so that's good, which is how to take the detailed models that we come up with and essentially sample them to be able to scale up to higher and higher densities and greater complexity. Which is something that the POC group is just touching upon with the strategy, but we definitely are looking for additional ideas there. So you imagine you see these microcarriers are 500 or so in that video that I showed. We of course need to get that up to hundreds of millions at least, and that computationally you can't just do that directly. So it seems like the scale up of detail to coarser large-scale models and longer-term models is most urgent in that discussion.
Cool. Thanks a lot, Simon. Okay, so I've got us randomized now. I think everybody, table captain should be right. So this is our next round. We have another 15 minutes, and I'll start it right now. So rooms are open.
Chris, we weren't randomized in our section. I don't know if everyone else had the same problem. Maybe. You're muted. You're muted.
Oh, I'm sorry, Elliot. That's too bad. I asked it to randomize, and the group I was in was different. So.
Oh, was it? Maybe ours was just perhaps.
Could have been bad luck.
Okay, that's what we were wondering, but I wasn't sure exactly.
Okay, we're back. Elliot, would you mind just sharing briefly one output that stood out to you from your session?
Sure. So there's kind of two main things that we talked about around what are the next parameters that might want to be looked at or included in the model, and what else might experimentalists want to capture in terms of the data that they're looking at that would best inform models. So some of the things that we talked about were adding in like power per volume for the bioreactor, the mixing time and the KLA constants, as well as the VVM, which I guess is a flow gases volume per volume of bioreactor metric, and the tip speed. Those are all important sort of parameters, and especially power per volume can inform scale up, I guess. And then we also discussed differences between, the expansion or proliferation phase and differentiation phases. So, could size and morphology of the cells, inform, how well an experimenter knows their cells are differentiated, for instance, without having to go and stain their cells or something like that.
This is the kind of stuff that we're going to be reflecting on. I'm going to ask table captains to report back to you at the end of our session. In the meantime, we've got a third round, and apparently one group was not randomized. I think you guys were amazingly randomized together again, so it should be different. I'm looking at the group mixes now. You should be really randomized. So round 3 is of the ideas you've discussed in the last 2 rounds, which one strikes you as the most important to carry forward now? And this is kind of like a little mini design session. So this is like, what consensus is it possible to come to in the next 15 minutes? If your table was gonna provide the impetus and the direction for this work moving forward, could you agree on what one step you would take, what one goal you would take for this work moving forward? So we'll open the rooms, and we have another 15 minutes. And then we're going to have a break, and then we'll move on to the last half. So rooms are open. You should be seeing invitation.
And we're back.
Chris, it
Chris,
looks like
are
you're
you
talking, but
talking?
you're muted. If you were talking to us.
I am. I was.
Okay, cool.
That was 30 seconds of brilliance that have been lost.
Oh, dang, missed it.
Oh, no.
So my invitation is to table captains to spend 1.5 minutes just summarizing the discussion that you had at your table.
So do you mean this last discussion? This consensus discussion?
Yeah.
Yeah, I think so. I'm happy to go first. I'll be quick, and I'm actually going to just share my screen because it's visual. Or I can't start. I can't actually permissions.
Oh.
Oh, Chris, can you just
you want to
unshare
share
real quick? Yeah, just it's visually, I think, useful. So I use this little whiteboard to capture stuff for the meeting. Briefly, we covered as this last talking point, it seemed like optimizing around cost reduction is a very umbrella goal that a lot of things can fall under. So we put that sort of at the top. And then from a first order of business, it seemed that validating the current model with additional experimental data that's either found in the literature or in partnerships would be super important as a first order of business. And you know, presumably hoping to get a predictive sort of model or outcome in place where you're then trying a new cell type that you might not have worked with before or used in your as the input for the model, and then having predictive power over that outcome. And then thinking longer term around the actual userability of these of the software, and how someone might want to be able to just go in and put in different parameters that they're interested in. So can we get to that point eventually? And then these were kind of two different large category buckets that I think are the next steps of or big areas of focus, but we didn't get into more nuance than that. That's my summary.
Thanks, Elliot. Who's next?
Can go next. So we had a discussion where we had two main strains of thought. The first was the academic side, where people want to incorporate as much biology as possible and make it as complex as possible. And the other side is the industry side, where people want results, want to know what shape a microcarrier needs to have have the best cell count, etc. So in the end, I think we decided that the biggest problem we need to tackle is how to sort of get these two sides together. Because as modelers doing this modeling, we do need the industry to validate our models. We do need experiments. But if the industry doesn't have time to do all of these experiments, which we need as modelers, which also involves doing lots of useless experiments which don't have results or have bad results, which are very interesting from a modeling perspective, but not from a commercial perspective, then it's hard to work together. So this is I think the problem we want to tackle first.
Thanks, Jaro. Who's next?
I can quickly go. So the consensus from our room was that validation, like other people have said, is the most important. You know, which includes, of course, incorporating more of the biological and biophysical aspects of the system into the current model, and then finding the right partner to run the validation experiments that are required to ensure that our model is accurate. And then the conversation took a bit of an interesting turn afterwards, from more of a social science perspective, and that's to whether or not we can get young people involved in this process generally, by, learning modeling at home, or, running certain simulations that can inform the current process, and so this is like taking advantage of the current COVID situation and, the next generation of modelers and hopefully cell-based meat consumers. So that's what sort of came out of our session.
Thanks, Greg. We have two more.
I can go next. So of course, being a high performance computing guy, I've been very, very focused on the scaling up thing and how to get to the full bioreactor, given that we're only looking at, thousands of cells. And I know we can't get past probably tens of billions of cells without too much computation. So that's what I brought up finally was, well, do we need to do this? And the answer was, well, really, no, that's not important. What's important is figuring out how to use the modeling to design better microcarriers. So that's been said already. That there's a big challenge in trying to understanding how we can redesign microcarriers so that we get more cells, more proliferation, and less differentiation. And again, to validate at smaller scales is really important to make sure that it is worth taking these models upscale, particularly looking at this whole range of behaviors from the cells when they're suspended to aggregating to adhering to microcarriers, because that's happening all the time in the bioreactors. Even though we usually talk about them being attached to microcarriers, they're not. And also by varying the parameters that are important for survival of the cells without getting into great detail, just trying to come up with some simple metabolics model that'll at least manage the oxygen, some key metabolic ingredients and key secretions. So really it's about validating and about coming up with better microcarriers is what I'm hearing as a consensus.
Thank you, Simon.
Maybe I can go next to close. Actually, there is a consensus, it seems, across all groups around validation of the model with more experimental data, and that came out as top topic also in our group. And there is more publications out there which we can use. And Steve mentioned a few of those which he authored, and there's a few others that he's aware of that we can leverage. But then we also concretely discussed about where can we do some of those experiments, and especially agitation came as one of the topics which probably will need to generate actually experimental data, because there might not be something in literature which is actually relevant. And A-star together with the Singapore group can potentially help with students to generate that. So I think that was a very concrete outcome. And the other topic was around the cells or the species that we focus on. We said that we want to be like species agnostic, but there was thinking in the group that actually it would be good that we maybe agree on some main principles when it comes to species. Like for instance, maybe we should pursue primary cells. It's like maybe non-negotiable for the group. And also deciding on a species that is like most ubiquitous and maybe also from a market opportunity, the most interesting one, maybe bovine, as a starting point, because the team felt if we start looking into some of the specifics of those species, it will make models so much more useful. But then of course there is a question, where do you start diverging from like, model that works for cross species for everybody, but then only starts working for a single species. So that was kind of it.
Great, thanks. Thanks, Dario, and great job, everyone. Thanks for being a part of that experiment. The documents we're gonna bring together, including things like the link that Elliot shared, and we'll be distributing that after the meeting so you can take a look at those notes and have those for your own reference. I would like to propose a 5-minute break. And when we come back, we're going to be looking at two things. One will be just the state of the CMMC as a body, its membership. One of the things, just as a preview, that we've been working on is how do we actually bring people together in a formalized way. So we've been working on some agreements and various things like that we need input on. And then also we're going to do 2 more rounds, just like we've done, but it's going to be expanding now beyond bioreactors, beyond microcarriers, and into just a broad inquiry into modeling in this field in general. So take 5 minutes. We'll be back at 11:02. Stretch your legs and we'll jump back in. Okay, so hopefully we're coming back from break, and if you are back from break, can you do me a favor and either turn on your webcam, just so I know that you're here, or go to participants, and there's a little icon. Well, there's several. There's a yes, a no, slower, faster, etc. You can raise your hand, do something like that, just so I know that you're here. Thank you. Still not seeing. There we go. Okay, great. It looks like most of us are back. Okay, thanks everybody. So I just want to take a very brief moment to share a bit about the design of the structure of the CMMC. And the reason this is important is that in the next several months, in our move to formalize the CMMC as an entity, and because we are working all together on generating new IP and new ideas, things like that, we realize that we need to formalize, and that includes membership agreements. And as we approached the challenge of how to build something that can really meet the needs of a very diverse group of stakeholders, we began to explore different kinds of models and agreements that could fit. And so what you're about to see is sort of a very quick overview of our current thinking. And all of this, I want to add, is up for discussion, and it's up to be shaped by this group. So this is not a final form, but I want to introduce where our thinking is at up to this point, and then also invite you to reflect on it with us and shape it together as a group. So just very briefly, the relationship that we have with content, with information, with a lot of stuff in our world is changing. And these next two slides come from our friends at guides.co, who have been thought partners with us on how to shape ecosystems, which is where our world is broadly heading. We started out as an analog world, we moved to digital. Right now we're in the process of seeing a lot of the rise of platforms, but now we're really starting new technology and frankly new cultural norms are starting to birth an ecosystem mindset. And largely that's going to shape and change how science is done potentially around the world. We're moving really from this competitive siloed pipeline, which is part of the reason why the CMMC exists. It's realizing that, especially in a field that's nascent and flexible and emerging, just like cultivated meat, we have an opportunity to come together and create a collaborative connected ecosystem to solve some of these problems that might be, if just one of us was working on them, we might arrive at similar solutions and it might take a lot longer. So what if we can come up with some shared solutions using a collaborative system? So when we think of the CMMC as an ecosystem, we realize that ecosystems really have three levels of system, very broadly. The first is the individual, it's each person on the call. The second is this community of practice, which is some among us are modelers, some among us are industry members, others are supplying the industry with products. Some of us have unique software approaches, etc. So, and there's also geographical cohorts and other ways that we can break up this ecosystem where there are people who have common purpose or a common skill set. And so the CMMC exists not just for the individual to get what they need in terms of being able to have access to different ideas or to provide value themselves through their skill set, but it's also a place for people to come together, discover those who are working on similar problems or have similar approaches, and to learn together, hopefully to begin to develop things like best practices or good practices, or to respond together collectively to changes that happen. And then finally, that next level is the ecosystem. It's really things like, how do you actually distribute ideas? How do you bring all of them together? How do you create an open system where new ideas and new energy can be continually coming in the form of insights, new technologies, new developments, things like that. And how do you, even on another level, for example, take advantage of what's happening in other fields. So regenerative medicine, for example, another place where you might see bioreactors. How is what they're learning and applying in that field potentially relevant to this one, and vice versa? So the chief question there really is just how do we generate value for stakeholders with different visions, concerns, business models, and scales? And it's not a small task. So how we are approaching that currently, our thinking with the CMMC is the goal is to keep membership a very low stakes proposition. So you should be able to join under an agreement of fair exchange and be exposed to the people, the communities, ideas, etc. At a very small cost, because every person who shows up on this call has different objectives and goals and things that targets and et cetera that they're working towards. So how do we enable involvement in the CMMC to kind of hit that sweet spot of you should be able to participate in a way that makes sense to you, that is in alignment with your other goals, and not be too burdensome. Yet at the same time, how do we support this community to be self-generating, for people to step into leadership roles, to be able to generate conversations and ideas and take projects forward? And then finally, how do we create mechanisms for multiple communities of practice to cross-pollinate this value? So there's two basic kinds of membership that we've sketched out, and there are two basic IP approaches. Well, it's one approach, but with two different branches. And the question here is, how do we create a fair exchange for stakeholders with all these different visions, et cetera? So the kinds of membership are based on fair exchange. And that first membership category are in-kind contributors, such as corporate and industry partners, who may join by contributing any combination of the following: funds that could contribute a problem space we really need to solve X, lab and equipment time, running validation experiments, introducing connections, marketing, blasting to various channels, actual labor, or paying for expenses. And then we have IP generators. And these are probably typically going to be people who are modelers and researchers and independents. And their agreement is to contribute the IP that they generate within the CMMC to the CMMC, so that we can maximize our purpose. And the IP agreements here are that all IP generated in the course of CMMC projects is released free for non-commercial use. And then second, to prevent the scenario in which we have a lot of different people owning and it not being very clear who owns the IP, we assign rights to distribute licenses to that IP for commercial use to the CMMC. And part of what these agreements support is a virtuous cycle. And when we started thinking about what does the CMMC as a system, as an ecosystem, every system is going to create these feedback loops. Some are homeostatic and don't change very much. They go up and down continually, and others sort of continue to run away. And we want to create this runaway flywheel of value. And so we thought about how do we actually map this out and think about it. And we won't spend a ton of time with this slide, but I just want to map kind of the major cycle. So starting at the top, we know that our purpose is that the cultivated meat market grows, and beneficially that the deleterious effects of factory farms are reduced. There's a whole bunch of other purposes that are tangential to that or fold into it. But basically, we think that this is a technology that is going to support the planet. So how do we make that happen faster? And so what we start with is this little cloud. The CMMC is bringing together funding sources. It's bringing together a diverse group of stakeholders together. For example, this group is this happening in real time. And from that cloud, we have two pieces. One is that the CMMC is going to generate IP, for example, like the POC that we've been sharing. And it's going to be generating this IP and both distributing it to researchers and academics who get to use it for free. And also there will be commercialization in some form and in some way. And that can look a number of different ways. The CMMC can commercialize that, or it can license others to commercialize that depending on opportunities that evolve and people who show up and raise their hand and say, I want to commercialize this. It's our belief that by creating this IP and making it non-commercial, free for non-commercial use, and also by creating these communities of practice where people will come together and they have these rich, meaningful discussions that actually move the industry and various skill sets forward, that we're going to be pushing through academic and research pipelines the kind of stuff, the information, the learning, the science that companies are going to need in order to make commercial products that are viable and to do it faster. And we also believe that if we are able to work with industry partners and actually commercialize projects like the POC, for example, and take them in a direction that is specific to either a subset of companies or to certain companies who want to use that tool, harness it, experiment, and move forward, that also is going to help and aid the design and optimization of products. All of that, which leads over to our left-hand side, companies bring products to market cheaper and faster. They don't have to explore this vast problem space with physical experiments. They can use modeling and information used for modeling. Cultivated meat is cheaper, more widely available, and we come back to our purpose. And then, of course, as the cultivated meat market grows, as this field expands, then more stakeholders are attracted to the field, which allows new, fresh inflow of expertise, funding, et cetera. So this is the virtuous loop that we're hoping to create. And I'm appreciative, there's probably a ton of questions too, but I'm just trying to give us as much of an overview as we can. So the structure of the CMMC, that's what's represented here by this little orange circle. What I'm about to share now is just some of the basic governance appliances that we have developed and been working with over the course of the last several months. Our structure is attempting to minimize barriers to participation, maximize leadership without the necessity of leaders, give members opportunities to steer the CMMC community, and then provide benefit to members both in their use of IP for their own purposes and for some monetary return should the CMMC license the IP. So we want to make sure that people who do show up in the CMMC and end up working on a project that is commercialized, that there's some remuneration for that above and beyond perhaps just the initial funding for the project. And our structure is guided by the principle that each entity or member should be able to participate fully without compromising their own individual purpose, as we are so diverse. So what we have, what emerged about 6, 7, 8 months ago was something that initially we called the working group, and we've now changed it to the steering group. And this was a subset of people who got involved in the CMMC, saw its potential as an entity and said, you know what, we really want to support this. Not only is it going to be helpful for us and what we're trying to do, but we want to put some energy and effort and time into helping this thing grow. And we've been meeting weekly. We look at the overall direction and steering of the organization, and that's the group that's come to you today and said, hey, we have a POC, we have this organizational structure, we have agreements that are starting to take form, etc. So this group is composed of members who are just taking an oversight role. It meets weekly. Decisions are made using some of the recent thinking that's emerged over the last 5 or 6 years around how do you actually distribute decision-making in a non-hierarchical structure. And that's where a lot of my own personal expertise comes from, and also Simon Cahan's. So we have an integrative decision-making process and some principles that we use that allow people to shape in real time suggestions for policies that they bring forward. And it's important to note that any member of the CMMC can make a proposal for a change. So if you're seeing a potential way to improve how we run the organization, or new structures or new roles or new means of organizing together, then anybody can make a proposal. And it's the steering group. You would be invited into the steering group temporarily to work on that. Joining the steering group at the moment, any member is eligible to join. And what we're asking is that people demonstrate a commitment to furthering the purpose of the CMMC. So it's this shift from I show up to the CMMC in order to work and collaborate with a group of people and work together on projects, to, well, now we're interested in working on the CMMC as a whole and seeing how we can support this thing in the future. Our current way to bring folks in is to vote them in. And we're very focused on really having that inclusion come down to, does this in any way hold us back from achieving our purpose? So purpose, we have to be rigorously focused on purpose in order to advance this thing forward. Otherwise, it starts to maybe take the shape of different individual contributors and what they want, what their need is. So we need to make sure that we're looking at what is the purpose of this body as a whole, and having that be our guiding principle. At the moment, we have a Cmmc Llc, and this is what enables us to form contracts and distribute funds, and it basically nests us inside of a legal structure. So the CMMC can hire administrators that perform a number of roles. They coordinate among members and stakeholders, schedule and facilitate meetings, maintain the budget, communicate to membership, perform outreach to partners. But it's important to note that in a traditional structure, usually it would be the LLC, the people within that, the ownership structure who have the authority. In this case, that body actually responds to what the steering group, how the steering group guides them. So I'm gonna skip this for a moment and jump in. We had a little bit different. I'm gonna talk about, oh, I think I missed a slide here. Okay. Well, I'm not sure exactly where that slide went, but let's just pause briefly, and in the chat, what questions do you have about the structure elements so far? What's the max size of the steering committee, right, Elliot? So I think this is actually a really good example of we don't know. So part of being a part of this new entity emerging is that we have a lot of things that we still have yet to decide. And our approach so far to this has been to say, what's the minimum structure that we need to create that'll help us move forward, and what's the minimum sort of the minimum viable structure that we need in order to answer the questions that could help us then answer the next questions. So I have no idea. Have we ever thought about incorporating as a cooperative or as a revenue generating nonprofit? Yes, absolutely. So this was a huge, this was a, the structure that's before you is the result of not only a lot of research and discussions that we had as a steering group, but also we've had a lot of conversations with people who are experts in various forms of organizing. And you know, there's trade-offs, there's pros and cons for a lot of this stuff. So what we actually ended up doing was deciding to incorporate it as an LLC, because that allows us to make shifts and changes much easier into perhaps alternative governing systems in the future. So this is really just keeps us flexible for now. And the invitation would be for those who are really interested in this kind of thing and want to be a part of ideating on how you do create a consortium that is future fit and evolves as an ecosystem, to look into that with us. Yeah, Simon, I'm going to call on you actually for Andrew's question. He asks, are there plans on how to handle IP considerations when universities and university labs are involved?
Yeah, we want to talk to the folks that are at those universities that handle their contracting. And we've done this with our GFI grant. We have an agreement in place with the University of Tennessee at Knoxville. And presumably different universities are going to have different structures and different ways of doing things. But we have a flexible enough setup in mind that we think it should be possible. So yeah, so if someone from a university wants to join, then we need to get in touch with the folks there that handle their grants contracting type office.
Right. We're going to touch on this in the end. This is the part that I was thinking I was going to talk about now, but at the end, we will be sharing that we have 5 different stakeholder groups that we've identified, that we've interviewed, and that we've developed some boilerplate template agreements for. And we'll be sharing those after the meeting so that y'all can peruse them, provide feedback, and if you want, be involved in helping take them forward and shape those as they move forward. So it'd be really helpful to get the feedback. One very helpful way would be to take that to your university and say, hey, this is the agreement that they're thinking about, and getting their input, helping us shape this. We realize it's a massive task to try to incorporate agreement that's going to work for everybody. So as Simon said, we're thinking about, we just realize that we have to maintain a certain amount of flexibility when it comes to negotiating those agreements between different groups that have different needs. Cool. So there's a ton of questions probably that come out of this that can be posted in the Slack. At the moment, I think we can shift gears now to the next round, which is really just opening up. We zoomed in on the POC, which is really great. Now it's time for us to sort of zoom out even more and think very broadly about what about modeling? What is the intersection between modeling and this field? What's the cutting edge intersection for right now? When we met a year ago, things were not as developed as they are now. So this is an opportunity for us. And again, one of the pieces of value that this ecosystem provides is bringing all these voices together to really reconsider what's the state of the art. So, so what is the next question, Chris? I'm glad you asked. So we're going to do 2 rounds, just like we did before. And the question is, what problems or challenges would benefit most from modeling? What kinds of problems would you be most excited to work on? So you can think about it from 2 different places. You can kind of take yourself out of the picture and think, what would benefit most as I look at this field and industry? Or you could bring to this question your own individual hat, your own framework, and say, what kind of problems am I most excited to work on? So any questions about that question or what our process is? I think we're all familiar now. I'm randomizing us.
Chris, I'd just say if with time considerations, we might want to set the limit to like 11 minutes or something like that per session, just so we can fit everything.
Okay.
Just heads up.
Okay, thanks. All right, everybody. So I'm going to open the room now. Now we're going to do things a little bit differently. Now, instead of having our table captains assigned, your first order of business is going to be to quickly decide who's going to be your table captain / note taker for this discussion. Okay, and opening all the rooms.
Okay.
So sudden.
Thanks for taking the notes, Leo.
Of course. Okay, so now looks like everybody's back. So now we're gonna move into round 2, and that question is, what next steps do we as a CMMC need to take in order to mobilize around these challenges? So I'm imagining that it's possible you came up with a number of really great ideas. You could pick something that you heard that felt really compelling, or you might have identified a bunch of different challenges, but maybe it's time to consider, well, just as a group, as a community, what next step can we all take together to move forward? It's up to you guys, whatever conversation develops. So I'm gonna randomize you again. Again, pick a note taker. Apologies if you get put in the same group. It should all be randomized. Okay, thanks everyone. See you back in 11 minutes. Okay, I think everybody should have gotten an invitation at this point. No, Leo? Okay, let me see. How about here? Pop up for you? Ah, that's weird. Okay, how can I force you to join? Let me see. Steve and Scott, I don't know if you guys can hear me. If you're having any problems, let me know. I can see if I can move you into the group manually.
Oh. I accidentally hit enter on the screen that had the button to return to menu instead of my notes screen. So I just left my group hanging.
Oh no. I
I
can't.
just have to say sorry.
I haven't figured out a way to actually put people into the rooms if they don't.
It's all right. I think there's only 30 seconds or so left.
Yeah. 12 now. How was the conversation? Was it
It was good. Yeah, I think this has been very well run, Chris.
Oh, good.
Well done.
Yeah, I'm really excited about the
Oh, to my group,
conversations.
Dario and others, sorry for leaving early. I accidentally hit enter on the screen that had the button instead of on my shared screen. So
Okay, we got something like that. No worries.
I think we're all back. Okay, so just a couple things before we wrap up in the next 10, 11, 12 minutes. There's a couple things that you can look forward to seeing very soon. One is that we need to take these formalizing steps. We have some contracts already in place, of course, and those are going to grow and expand. So you should be able to look for a prototype formal membership agreement. It'll probably be 5 because we have 5 different stakeholder groups. Those will arrive in your inbox. If you could, please consider sharing them internally with people who might be able to support from your organization, giving some feedback on that agreement, both feeding back to you, how that would jive with their own structure, et cetera, and also to be able to feed back to the larger community what's useful. Our goal is to begin moving towards a formal membership body as we receive that feedback. And hopefully we can complete that by the next general meeting, which we have tentatively pegged as November 2020. It was going to be in person, of course, but looks like we'll have to do another one of these. And then when it comes to the POC and beyond, of course, our goal really is to extend this work into new project teams and features. So we have the work that's currently moving forward, but there's an opportunity now because of this community and what's already been created to expand that. So the POC will be moving into another phase of development that will begin September, working on the GFI grant. We'll be bringing in a couple new folks who have been hired specifically for that work. And I would ask that you consider inviting fellow members in Slack to continue the discussions that we started here today. We have a Slack community. If you're not a part of it, that we can easily invite you. There's an opportunity here to start channels, start conversations, move things forward in an asynchronous way. And just ask yourself what new applications and features you might be interested in exploring or providing feedback to those who are moving those things forward. And so just to prime your thinking a little bit, you know, that I think that there's always the danger, you know, of we showed up and we said, hey, here's what we have done, and we're presenting it to you, right? And our really, as an ecosystem, our goal ought to be that the we incorporates more and more and more of us. But it needs to be, as we've said a couple times now, in a way that's elegant, fits into your current working styles and arrangements and things like that. So in a minute here, just to check out, I'll ask people to reflect on how they want to contribute or show up or what they might need to do that. But here are just some suggestions. You can invite others who are aligned with our purpose to check out the website, forward them that stuff. If you're a member of Slack, you can start a channel, join a discussion, or pose a question. If you're not a member, we can invite you. You can invite somebody who's on the steering group to share more about the CMMC in depth if you have more in-depth questions. You can consider inviting people to a presentation of your work. So you can consider inviting a subset of us or all of us to say, hey, I want to explore and explain some of what I'm interested in and see if other people want to get a conversation going on that. You can call for an online meeting for members to gather around a particular topic. And these are Oh, and Elliot brings up a really good point. Most of our membership is male. Think about your female colleagues, other diverse backgrounds who might be interested in the CMMC. We need to grow, we need to become more diverse. And yeah, so these are just some suggestions. Now we have an opportunity just to check out if you already consider yourself to be a member of this community, what next step will be most impactful for you to take in the coming weeks? And for those who aren't members, but maybe you're cheering us on from the sidelines, or you're not sure if you want to be a member yet, what next step can this community take to support what matters to you? What ask would you make of us? And we're just going to do this as a checkout. So how I'm going to do that is just go in order and call on folks at random. And just keep your answer to maybe anywhere between 30 seconds to a minute. And then we'll wrap up. So I'll just give you a few seconds to ponder the question. And as you're pondering, I really want to thank everybody for showing up. The energy that came out of our June meeting was incredible. The relationships, it's been amazing to create a community of colleagues and to see so many familiar faces and new faces. I just really want to thank you for being with us for 3 full solid hours in which a lot was called on you to participate in these discussions. We really appreciate it. I do. Okay, so, one last thing, sorry, before we get going. Could you please send us your notes? You could do that a couple different ways. info@thecmmc.org. You could reach out to us directly, post them in the Slack. Just find some way to share your notes, especially table captains, please. All right, so I'm gonna just start calling on people. The order that I have on my screen is probably not the same one you have, so just be ready. And we're gonna start with Elliot.
Okay, well, for me, if people that don't know me, I'm the senior scientist at GFI, kind of specialized in cultivated meat. So I think for me, some of the most important things are helping to help prioritize what projects should be worked on, and sort of thinking about what's going to be most impactful for the industry, and taking all this sort of stakeholder involvement and conversations that I have on a day-to-day basis, and bringing that into the CMMC and hopefully informing what we do. And then in addition to that, using GFI's network and my own network to get the word out.
Thanks, Elliot. And Elliot is a member of our steering group. And then next we have Nico, and after that, Patrick.
You're muted, Nico.
Thank you.
Thanks, Nico.
Not much to add anymore after 3 hours. To be honest, I found it quite valuable, and yeah, I like to see how the CMMC follows up actually with practical models. Well done.
Thank you, Nico. Okay, Patrick, and then Simon.
Okay, yeah, for me, the thing for me to impactful for the next bit is to look out and see if there's other collaborations that can start to form on projects. Are there opportunities for other type of funding, or ways that we can work together to bring some piece or some component to fruition that can assist in the goals of the CMMC.
Thanks, Patrick. Simon, and then Leo.
Yeah, most impactful. That's always tough. Extreme. I know I need a rest. That'll be one thing that'll be impactful. But I'm wondering about what projects to pursue next, to propose next, and I've got some ideas from this discussion. Modeling the shapes of microcarriers and the effects that can have, and so on. I'm interested in reconnecting with people that would like to pursue joint project proposals. So please get in touch with me if that's of interest.
Thanks, Simon. Leo and then Netta.
Thanks, everyone. I'm Leo. I really put myself in the bucket of potential members more than current members. I think for me, the most useful thing would just be to have ways of finding how I can help out. As someone kind of from the outside looking in on this work who really wants to help and find a way to contribute, I think things like job boards or even Slack channels that sort of function a little bit as like job boards or ways for people to look for ways of plugging in. Even just meetings like this give me the opportunity to see the kind of work that's happening. So just more of the same, I suppose. Thanks, everyone.
Thanks, Leo. We'll go Netta and then Ludwig.
Hi, thank you. Sorry for joining late. I had other obligation and I really enjoyed it, and I was very glad to see the progress that the society went through in the last year. Unfortunately, I was too busy to join, but I think one of the things that as a startup that is very, very busy with all kinds of missions we can contribute is really to publish, communicate about the establishment of the community. We participate in many conferences, and we can of course add a slide that publicate, for communication about the CMMC. And of course, take a part as a member, because now it's much clearer to understand what is the legal issues around participating.
Thanks, Netta. Ludwig and then Arya.
Yeah, this was great. I think for me, as a quite new person to this field, some entrance level tasks based on like skills and interest, like you can, I think I did it when I joined the Slack group, so that you're interested in, but if there's a more structured way to find ways to contribute and slowly, on the side of doing studies and everything, contribute with minor tasks or longer projects maybe. Yeah.
Thanks, Ludwig. Arya and then Carol.
Yeah, I just wanted to say thank you. I know we're running short on time, and it's really amazing to see all the progress. I think that creating more communication in the next few weeks around, I think, alignment of mission, we were talking about this in one of the small groups, is I think already there with a lot of the startups, and maybe, like I would just say also just, prioritizing those projects and kind of, addressing how that looks and a sort of, timeline for these things would be super helpful.
Thanks, Arya. Carol and then Jaro. I'm just noticing Carol, while you're finding the mute.
Sorry. Yeah.
I noticed that I noticed we're over time. So if you got a jet, totally understand. If you could just answer that checkout question for the sake of everybody in the chat, we'll say goodbye. Otherwise, if you could stick around for just a few more minutes, it'd be great to hear some more voices.
Okay. So thank you so much. The meeting was awesome. I learned a lot. And I think I'm more curious to see the difference on the models of the different bioreactors. I really want to hear the outcomes of that, and how is the validation of the models and the real experiments, and how this could impact the startups.
Thanks, Carol. Jaro and then Dario.
Right. I think I have two things. A personal next step will be to maybe try to get a group together to dive more into the biology and go into really dive in deep into the science of modeling these cells. But on the other hand, I think one of the discussions and the conclusion that we align the goals for the commercial partners and the academic partners, which might be a problem if the academic ones dive in deep into the science. So how we're going to do this, it's going to be an interesting question, which I want to answer. And now I'm signing off to feed a baby.
Okay, happy baby feeding, brother. Dario and then Rob.
For me, the most interesting will be actually to synthesize the information that came out from this workshop. I think it was an amazing session, great feedback from many people, and now bringing it together and defining the work packages so that everybody can contribute in a meaningful way for kind of CMMC's benefit, but also for their own benefit. I think that will be a very interesting piece of work, which will hopefully bring us all forward.
Thanks, Dario. Rob, and then Andrew.
Well, thank you so much, folks, for inviting me. I think this community is absolutely incredible. I'm in awe and humbled. My work, currently working with Elliot on an NSF grant that would be multi-institutional. I think thinking of ways to align incentives, as some folks were talking about earlier, or align mission between the different university projects and what CMMC is doing, and thinking about ways to use digital tools, I think would be really neat. What could the community do to support me? Hopefully keep inviting me back. I have a under review where I talk about participation in cellular agriculture. I talk a little bit about what CMMC has done in terms of the white paper and its goals. And I'd like to be more engaged in that type of conversation going forward as well.
Awesome, thanks, Rob. Go to Andrew and then Clarice.
Okay, to echo Rob, thank you for hosting this and having me. It was great to see old faces again, meet some new faces. I thought it was productive and always interesting discussions, and I'm definitely not a modeler myself, so I always learn a lot with the CMMC. And that ties to, I think, kind of where I can potentially be the most valuable is just on the experimental side of things. So I think that what I guess I'm doing right now is just saying that people can feel free to reach out about collaborating on the experimental side of things. I'm part of a lab group at Tufts that we'll have 5 people working dedicated, 5 PhD students dedicated to cultured meat research this coming year. So we have a pretty big group, and I think have potentially a lot of capacity for helping out. Mostly I'm speaking of my colleagues because they're the smart ones. Yeah, so thank you.
Thanks, Andrew. Going to Clarice and then Sagit.
Hi, so thank you. It was so nice to get some ideas because I'm planning to embark on a PhD journey as well. And that's why I think most important for me would be to know how we're moving forward, like urgently how to collaborate and in terms of timelines, as we said, and funding. Thank you.
Thank you, Clarice. Sagit and then JiaJia.
Hi, thank you. This field is actually new to me. And I will say that the important thing is the collaboration, the supporting of the model with the experimental data.
Thank you. Thank you very much. JiaJia, you're next, and then Maria.
Hi, everyone. I'm JiaJia. So I think for the next step, definitely is continue to work the funded projects, and even I'm actually new to this area, but for the project, we kind of recently hired new graduate students. So the next step is to educate even younger generations of modelers to kind of make a big contribution to this area. And also I would like to identify more collaboration opportunities with all of you to see future more collaborative work on this.
Thanks, JiaJia.
Thanks.
Maria and then Tim.
So thank you for this meeting. It's my first experience in such kind of meetings, and our company is important for our company. It really matters to receive any information that you can give us. Thank you very much.
Thank you. Thank you. Tim, Greg, and then Dimitri to finish us off.
Oh, this has been so much fun. I've loved every minute of it. I'm really excited to help out with the social media, being able to get the information that the CMMC has out to everyone so everyone can stay really with their finger on the trigger of all the information is something that I'm really excited to get to help out with.
Thanks, Tim. Craig and then Dimitri.
For me, it's just trying to sort of synthesize all the feedback that we got from people to sort of ensure that the work that's going to happen next with the proof of concept project and beyond is as relevant to what people need in industry as possible, you know, and to sort of figure out new ways to translate that kind of, you know, science that's done in industry into sort of computational modeling. So sort of connect the dots between those two things. That's what I feel is like the way that I can be most impactful in the short term.
Awesome, thanks, Greg. Then Dimitri, you get to round us out.
Thank you all. It was a good experience for me because I don't have a large experience in this area, and I like to listen to people who get more engaged in this area. And moreover, I want to thank the organizers of this webinar, if I can say so, and I hope we will see each other again next time. Thank you all.
Awesome. Well, thanks everybody for coming and giving us all that time and for staying a little bit extra longer to say goodbye to everybody. We'll send out all the stuff we promised, and hopefully we'll see you on the Slack and in future meetings.
Yeah, thanks, Chris, for running this. Appreciate it.
My pleasure. Bye, everybody.
Thank you, Chris.
Thank you. Thank you.
Bye.
Thank you.
Thanks all.
Thank you very much.
Thank you.
Bye-bye.
Bye.